Today we will discuss about the DNA replication. What is DNA replication? DNA replication simply means the synthesis of DNA. DNA replication means synthesis of DNA. So DNA replication is the DNA dependent DNA synthesis. What is DNA replication? DNA dependent DNA synthesis. DNA synthesis. DNA replication is DNA dependent DNA synthesis. That is, when DNA synthesizes itself, that is what is called as DNA replication. So we have got DNA and it will synthesize itself, that is called as DNA replication. It's called as DNA replication. This DNA replication, it occurs during the S phase of the cell cycle. That means it occurs during the synthetic phase of the cell cycle. Synthetic, what is called as S phase of the cell cycle. The DNA replication, it occurs during the S phase of the cell cycle, what is called as synthetic phase of the cell cycle. Now, what happens during the DNA replication? See, we have got a parent DNA molecule. For example, this is parent DNA molecule. Obviously, this is sugar phosphate backbone. This is sugar phosphate backbone, and inside the helix, nitrogen bases are present. Inside the helix, nitrogen bases are present. These are nitrogen bases which are present inside the helix. DNA replication means that in case of DNA replication, this DNA molecule, this is called as parent DNA molecule. This parent DNA molecule, it unwinds the two chains of the DNA, this parent DNA molecule, they separate, and then each chain acts as a template for the synthesis of new DNA strands. It will be like this. For example, this is parent DNA molecule. Parent DNA molecule. Then it will undergo DNA replication. This is parent DNA molecule. In DNA replication, what happens? This parent DNA molecule, it has two strands. We know this is one strand, this is another strand. These two strands separate, and each strand acts as a model or template for the synthesis of new DNA strand. It will be like this. This is one strand. This is another strand. So these are two parental strands or old strands. Now new DNA strands will get synthesized. These are new DNA strands. So this is DNA replication. DNA replication means that the parent DNA molecule forms two daughter DNA molecules. They are called as daughter DNA molecules. And in these daughter DNA molecules, this is one strand. This strand is one strand is old strand, old or parental strand, old or parental DNA strand, and another is newly synthesized DNA strand, and this is newly synthesized DNA strand. This one is newly synthesized DNA strand. This is newly synthesized DNA strand. So what do you mean by replication? Replication is the DNA dependent DNA synthesis. That means for the sake of replication, this DNA uses the sequence and then synthesizes its own DNA. That is called as DNA replication. DNA dependent DNA synthesis. During the DNA replication, the two strands, we know that DNA molecule is double stranded. The two strands of the parent DNA molecule get separated, and then each strand, it acts as a template, it acts as a model for the synthesis, for the formation of new DNA strands. And this is called as parent DNA molecule, and these are called as daughter DNA molecule. So in a daughter DNA molecule, we find two strands. One strand is old strand or parental strand, and one strand is newly synthesized strand. These two daughter DNA molecules, they are exact replicas of parent DNA molecule. They resemble with one another as well as they resemble with the parent DNA molecule. So these daughter DNA molecules are exact replicas, or called as replicas, carbon copies of parent DNA molecule. Parent DNA molecule. So in replication, two daughter DNA molecules are formed, and these daughter DNA molecules are exact. They are exact replicas, carbon copies of the parent DNA molecule. See, these daughter DNA molecules which are formed here, these are identical to each other. They resemble with each other as well as they resemble with the parent DNA molecule. This is what happens in case of replication. So I told you that the newly synthesized daughter DNA molecules, they have one old strand and one new strand. This type of replication is called as semi-conservative DNA replication. So semi-conservative DNA replication, semi-conservative DNA replication. Semi-conservative DNA replication. Semi-conservative DNA replication means that it is called as DNA is half conserved. What do you mean by semi-conservative DNA replication? DNA is half conserved. It means that in DNA replication, in case of daughter DNA molecules, two strands are present. These are daughter DNA molecule, two strands. This is one strand, this is another strand. Obviously, one strand is parental strand and another strand is newly synthesized strand. So we say that this type of replication is called as semi-conservative mode of DNA replication. And this semi-conservative mode of DNA replication is that type of DNA replication in which half of the DNA is conserved and second half is synthesized anew. That is called as half of the DNA is half conserved and second half of the DNA is synthesized anew. That is what is called as semi-conservative mode of DNA replication. One strand is old strand and one strand is newly synthesized strand. We call this type of DNA replication as semi-conservative DNA replication. So then we'll talk about the semi-conservative DNA replication, in which one strand is older strand and one strand is newly synthesized strand. That is, DNA is half conserved and second half is synthesized anew. That is called a semi-conservative mode of DNA replication. This was first experimentally proved by Meselson and Stahl. So semi-conservative mode of DNA replication was first explained by Meselson and Stahl. And this Meselson and Stahl, it provided evidence for semi-conservative DNA replication, for semi-conservative mode of DNA replication. It was an evidence for semi-conservative DNA replication. And these Meselson and Stahl, they explained that DNA is half conserved in Escherichia coli. The experimental stuff was Escherichia coli. We often use bioassay. We say Meselson and Stahl's bioassay. Bioassay is simply an experiment which is related with the living organisms, experiment dealing with living organisms. So we can also here write Meselson and Stahl bioassay. It is written in certain books. Then we have got what this Meselson and Stahl's experiment. Meselson and Stahl's experiment, he used these two scientists, they proved that DNA replication is semi-conservative. We'll discuss what their experiment was and how it proved that DNA replication is semi-conservative. They experimented with Escherichia coli, E. coli. They used two isotopes of nitrogen for their experiment. One is heavy isotope of nitrogen. There is Meselson and Stahl. They experimented with Escherichia coli, and they used two isotopes of nitrogen. One is heavy isotope of nitrogen, that is N15, and normal isotope of nitrogen, which is light normal isotope of nitrogen, and that is N14. Both N14 isotope of nitrogen and N15 isotope of nitrogen are stable isotopes of nitrogen. These both N14 isotope of nitrogen and N15 isotope of nitrogen, they are stable isotopes of nitrogen. Then they used also in their experiment. What they used the cesium chloride density gradient centrifugation technique. They used cesium chloride density gradient centrifugation technique. They used the cesium chloride density gradient centrifugation technique. This is cesium chloride. This how this experiment proved that it is the DNA replication is semi-conservative. Actually, this cesium chloride solution, this cesium chloride solution, it shows density gradient. That means it shows density gradient. That means it forms on centrifugation. It shows density gradient. That means on centrifugation it forms bands of a solution of lower density and bands of a solution of higher density. It would be like this. For example, we have this. We have got here. We have got cesium chloride solution. This is cesium chloride solution, and this is centrifuge tube. This cesium chloride solution, it shows density gradient. That means it shows the bands of higher density at the bottom and bands of lower density at the supernatant. That means this is called a supernatant. This is supernatant. Here we find the cesium chloride solution which has low density, which has lower density, and here we find stuff which has higher density, which we find cesium chloride solution which has higher density. It is called as pellet. So we can say that it is having higher density solution, and it is having lower density. What I mean to say is that the solution of cesium chloride which is in the pellet, that is having higher density, and the solution which is present in the supernatant part of the centrifuge, that is having lower density. So they used this cesium chloride density gradient. Gradient means difference. That means there is difference in density. The solution which has lower density, that will be in the top, that means in the supernatant, and the solution which has higher density, mass per unit volume, that means that will settle in the bottom, that will be in the pellet. That means it is a bottom. Or we can say that molecules which have higher density, mass per unit volume, they will settle in the bottom. That will be the pellet part, that is higher density. And those molecules which have lower density, which have mass per unit volume is lower, they will be in the top. They will be in the upper part. That means they will be in the supernatant. This is what is called as density gradient. That means difference in the density. We can explain like this. For example, if this is I will show it like this. This shows density gradient, cesium chloride density gradient. That means at the bottom in the supernatant, density is greater, and in the supernatant, in the pellet pot at the bottom, the density is greater, and in the supernatant or in the upper pot, the density is lower. So these thick line indicates that higher density. This is higher density and this is lower density. Higher density is in the pellet or in the bottom, and lower density, molecules with lower density, they would be in the upper pot or in the supernatant. This is what is called as density gradient. That is difference in the density. It is as simple as this. Those molecules which have higher density, which have higher mass per unit volume, they will settle in the bottom. That is what is called as pellet. And those molecules which have lower density, they will be in the supernatant, that is upper pot. So if you consider that this is the bottom and this is the upper part. The denser molecules will be in the lower part, that is in the pellet, and lighter molecules will be in the upper part, that is called a supernatant. This is what is called a cesium chloride density gradient. In this experiment, what they now they want to prove that DNA replication is semi-conservative. If we'll just put DNA molecules of different densities in this cesium chloride solution, if we'll put the DNA molecules of different densities in the cesium chloride solution, we find that these DNA molecules will separate, and these DNA molecules will separate according to their densities. That means heavier density molecules, DNA molecules with heavier densities, they will form a sort of gradient along with the cesium chloride solution. That means they will heavier DNA molecules, they will be in the bottom, that is in the pellet, and the lighter DNA molecules, they will be in the upper part, that is in the supernatant. This is the basic crux. This is the basic what I can say, that the basic principle regarding the semi-conservative DNA replication. That means when DNA molecules of different densities, they are mixed with cesium chloride solution, they form density gradient. That means they would separate and form the density gradient. That means DNA molecules with higher densities, they will settle in the bottom, that will be in the pellet, and those DNA molecules which are lighter, they will be in the upper part, in the supernatant part of the centrifuge, and that is, they have formed the gradient along with the cesium chloride solution. That is why it is called as cesium chloride density gradient. Now what they did, what was their experiment? They took an Escherichia coli. This is first they made an culture medium. This is culture medium. In their experiment, let me first explain what they did in their experiment. In their experiment, they made bacterial DNA, heavy DNA, and bacterial DNA, light DNA, and bacterial DNA, hybrid DNA. That means the bacterial DNA was made heavy by means of heavy isotope of nitrogen. So they made a culture medium. In this culture medium, bacteria with the heavy isotope of nitrogen, bacteria were cultured in a heavy isotope of nitrogen, in a culture medium having heavy isotope of nitrogen. That means in this culture medium A, bacteria cultured in ammonium chloride solution. But this ammonium chloride solution, here nitrogen was heavy. That means the bacterial DNA will incorporate heavy DNA. The bacterial DNA, these bacterial DNA would be having nitrogen bases which are having heavy isotope of nitrogen. That means N15. So we can say that this is culture medium. This culture medium is having heavy isotope of nitrogen, N15 isotope of nitrogen. Have N15 isotope. This is heavy nitrogen, and bacteria were cultured in this culture medium having heavy isotope of nitrogen. Actually, this culture medium contained ammonium chloride, and this ammonium chloride was having what? Heavy isotope of nitrogen in it. So the bacterial cells bacterial cells, they became heavy. How they became heavy? They incorporated heavy isotope of nitrogen in their nitrogenous bases in their DNA, and we say that their DNA became heavy. So it would be like this. So these are the these were the bacteria having the heavy isotope of nitrogen. This is heavy isotope of nitrogen. Heavy isotope of nitrogen. Then what they did, they tested these bacterial cells by means of centrifugation. So they tested this DNA or these bacterial cells by means of centrifugation. What they did, this it is having this on centrifugation, on testing the DNA with centrifugation, they found that this DNA formed band here. DNA formed band here. So this is heavy DNA. It is a band of heavy DNA. Band of heavy DNA. It is a band of heavy DNA. So we can here write it is a band of heavy DNA. Heavy DNA. This is obviously cesium chloride. This in the cesium chloride solution, this DNA formed a band at a particular location in the pellet, that is called as, that is in the pellet this heavy DNA formed a band. So we can say that this is cesium chloride density gradient, cesium chloride density gradient. This is cesium chloride. This is centrifuge tube. This is centrifuge tube. Obviously these bacterial cells would have heavy DNA. Then what they did, they transferred in the first part, they cultured bacteria in the heavy isotope of nitrogen, and their nitrogen bases incorporated heavy isotope of nitrogen, and then on centrifugation they tested this DNA by means of centrifugation, and in the cesium chloride density gradient, in the cesium chloride in a centrifuge, this heavy DNA, it formed a band in the pellet pod, and this band constitutes the heavy DNA. That means DNA having the heavy isotope of nitrogen. So in the next part, what they did, they transferred these bacteria from N15 culture medium to N14 culture medium. Then they transferred these bacteria to N. They transferred these bacteria to N. This is another culture medium having, this is culture medium, but this is having N14 isotope of nitrogen. N14 isotope of nitrogen. Actually this culture medium was having what? Ammonium chloride solution, and this ammonium chloride solution contains light isotope of nitrogen, your normal isotope of nitrogen, that is N14. So these bacteria were transferred to, this is bacteria transferred to N14 culture medium. They transfer to N14 culture medium. And after this, when they were transferred to N14 culture medium, after 20 minutes is the generation time for the E. coli. That means it is a time after which this DNA divides. That means this bacterial cell divides. So after 20 minutes, what they found? After 20 minutes, they got the first generation. This is after 20 minutes. This is 20 minutes. This is generation first. These bacteria were transferred to N14 culture medium. Agreed? And then after 20 minutes, this they got the first generation of the bacteria. This generation time is the time when the bacterial cell divides, and that means it is DNA replicates once. The generation time for the E. coli is 20 minutes. 20 minutes. That means this generation time for E. coli is 20 minutes. That means after 20 minutes, this bacterial cell divides and it is DNA replicates once. After 40 minutes, it will form second generation. So we can say that after 20 minutes, they got the second generation of bacterial cells in the N14 culture medium, and they tested the second generation of bacteria by means of centrifugation. And what they found? To their surprise, they found the hybrid DNA. They found what? They found hybrid DNA. And this hybrid DNA, it formed a band a little bit higher as compared to the band of the heavy DNA. This heavy DNA is formed band here, but this is the band of heavy DNA. But this DNA, hybrid DNA, it formed a band a bit higher in the cesium chloride solution. This is hybrid DNA, cesium chloride solution. This is cesium chloride solution. This is cesium chloride, and this is the hybrid DNA. This is the hybrid DNA. Hybrid DNA is the DNA which is intermediate. This hybrid DNA is a DNA which is intermediate between the heavy DNA and light DNA. So hybrid DNA, it is an intermediate between the heavy DNA and light DNA. See, after first generation, after generation time of 20 minutes, the DNA of these bacterial cells were tested on by means of centrifugation in cesium chloride density graded centrifugation, and then they found that this DNA formed a band a little bit higher as compared to that of the heavy DNA. So they called this DNA as hybrid DNA, and this hybrid DNA, it is having two strands. One strand is N15, another strand is N14. While as in case of heavy DNA, it is both strands are N15. So a little bit you have understood that one strand is older strand, parental strand, one strand is new strand. So this is called as hybrid DNA. And after second generation time, that means after 40 minutes, what they found? They found two DNA types of DNA molecules. One is hybrid DNA, and another is what? Another is this light DNA. This is hybrid DNA. It's hybrid DNA, and another is light DNA. Another is light DNA. So two types of DNA molecules were found in the generation second. This is generation second. So we have got two types of DNA molecules. One is, this was hybrid DNA, and this is what is called as light DNA. Obviously this light DNA, it is having what type of nitrogen? It has two strands. Both strands have N14 type of nitrogen, normal isotope of nitrogen or light isotope of nitrogen, that is N14. This is having N15, N14. So obviously this is, if we'll take this bacterial cell, this is called as heavy. This would be heavy. That means one strand, this is one strand, this is another strand. So it would be N15. So it is called as heavy DNA. This is bacterial cell. This bacterial cell, this is, we have cultured this bacteria in heavy isotope of nitrogen. So bacterial cell would have two strands. Both strands are what? Both strands are having N15 isotope of nitrogen, and it is called as heavy DNA. Then here we have bacterial cell. But this bacterial cell is having an intermediate DNA or hybrid DNA. This is one is N14, another is N15. So this is what is called as intermediate DNA. This intermediate DNA or hybrid DNA. This intermediate DNA, it is having the density in between the heavy DNA and light DNA. So, and this intermediate DNA or hybrid DNA, it has two strands. One strand is having N15 isotope of nitrogen and another strand is having N14 isotope of nitrogen. So this is called as intermediate DNA or hybrid DNA, which is having density intermediate to N15 and N14 type of DNAs. Then it is the bacterial cells. This is called as these bacteria in the second generation. We would get the bacterial cells which would be having light DNA. That means light DNA means N14. So light DNA, it has two strands, N14. And also we will get what? Bacterial cells which have intermediate DNA. So bacterial cells which have intermediate DNA. This is bacterial cell. The bacterial cell which is having what? It is having hybrid DNA, that is N14, N15. So it is called as intermediate DNA or hybrid DNA in the second generation, hybrid DNA in bacterial cell. So this experiment has clearly shown that DNA replication is semi-conservative. DNA replication, there is semi-conservative mode of DNA replication because of the fact that when you have taken this heavy DNA, in a culture medium, then you have transferred the bacteria in the another culture medium which is having normal isotope of nitrogen. When after first generation, these bacterial cells with heavy DNA, they have started replicating and they have separated their strands. In this case, they have separated their strands after 20 minutes, after first generation time. After 20 minutes, they have separated their strands and they have replicated. Now in replication, they have got one parental strand, one older strand, and one newly synthesized strand. And that nitrogen they have got from this nitrogen which is present in the nitrogen base, they have got it from the N14 culture medium. As a result of that, we obtained hybrid DNA or intermediate DNA. Formation of hybrid DNA or intermediate DNA when we take the heavy isotope of nitrogen and light isotope of nitrogen, it clearly indicates that one strand is parental strand, old strand, one strand is newly synthesized strand. This clearly indicates that DNA replication is semi-conservative mode of DNA replication. This was first experimentally proved by Meselson and Stahl using the heavy isotope of nitrogen N15 and N14 and using the cesium chloride density gradient. Why they used the cesium chloride? Because by and large the density of the cesium chloride is almost the same as that of the DNA. That is why they used the cesium chloride density gradient centrifugation technique for their experiment to determine that it is the DNA replication is semi-conservative. Thanks.